thank you for that kind introduction, and thank you all for coming here today to the conference. My name is Olivera, and I'm the COO at Olivera Mitevska, where we do genetic testing for neurodevelopmental disease, as well as oncology. So today I want to talk to you about the role of whole genome sequencing in personalized treatment approaches for autism spectrum disorders. Now that's a lot of words, and we're going to break them down in a much simpler way. So the main two takeaways from today's presentation should be, first of all, why is personalized treatment necessary for autism? And secondly, how can genetics, specifically this genetic test called whole genome sequencing, help us in achieving this goal? So we've heard a lot today about what autism individuals face socially, but let's shed a light on the medical comorbidities that they may face. So beyond the core features of the social communication impairment and the repetitive or restricted behavior, autism individuals can face a variety of comorbidities from seizures to sleep disorders, asthma, allergies, different food sensitivities, as well as psychiatric symptoms such as ADHD symptoms, anxiety, depression, OCD-like symptoms, bipolar disorder-like symptoms, as well as GI, gastrointestinal symptoms like cycles of constipation and diarrhea, or just constipation and diarrhea, reflux, and abdominal pain or IBS-like symptoms. And this is a non-exhaustive list of all of the comorbidities. Maria will share them more in the next presentation. So really, autism spectrum disorder is not purely a neurological disorder if we look at it holistically, because it affects the whole body. It's a system disorder, and we need to treat it accordingly. There are a variety of treatment options for patients with ASD, all tackling it from different angles. We have speech and language therapy, occupational therapy, behavioral therapy, as well as some new and emerging assistive technology devices. We have alternative approaches, such as the puppy play that we heard about, and we also have psychological and mental health therapy. But today, I'll be talking about the medical and pharmacological aspects of autism treatment. So here's a list of some commonly prescribed drugs for the comorbidities associated with ASD. And why do I say comorbidities? It's because to this date, there's not an officially approved treatment for the core autism symptoms. We have risperidone and aripiprazole, which are atypical antipsychotics that have FDA approval, but only for irritability associated with autism. And very recently, on September 22nd, the FDA approved leucovorin calcium, but only for a specific subset of patients who have cerebral folate deficiency associated with ASD. And maybe Dr. Frey will talk to us about that a bit later. So here we can see this is again a very non-exhaustive list of the medications that may be prescribed to autism patients. And as you can see, it includes a variety of things from mood stabilizers to antidepressants, anxiolytics, and as well as some medications that you might not consider are relevant for autism, like GI medications or heart medications that your grandmother may be taking. So there's no shortage of options, right? So why does then autism continue to pose a problem for so many individuals? Well, if we look at the current ongoing clinical drug trials, we'll see that there's, again, no shortage of option, and this is just a sample that I came up with. There's hundreds of studies being done right now, and they're all tackling autism from different pharmacological approaches. But now we'll focus on a case example, bumetanide. So, what is bumetanide and how does it work, or how is it proposed to work? It inhibits a certain calcium channel in the brain that is thought to have a very high concentration of intracellular chloride. Sorry, it's a sodium-calcium chloride transporter, but it's thought to inhibit this transporter so that the chloride ions, which are in very high intracellular concentration during early brain development, are lowered, and that the balance between excitatory and inhibitory signaling through GABA is restored. So I won't bore you anymore with the complex medical details. Why is this like here? Because there was a big study, phase 3, which is towards the final stages of the clinical trial pipeline, and it was terminated. Why? Because of a lack of efficacy. But then there are other smaller scale studies, even this one which came out after the study was terminated, which showed that in a smaller cohort, this medication was effective for some kids. So why is this medication effective in some kids and not in others? Well, we can think of every autism patient as a unique person. That's why I have fingerprints here, because we're all unique. And not everyone is going to respond to the same medication in the same way. Now, because of these differences, some people might be better suited to bumetanide or some other type of medication, but when placed in a large cohort, these sizable effects get obscured. So what is the reason, or what could be the reason behind these differences in patients? Well, we have to look at the potential causes of autism spectrum disorders. Now, as you all know, there's no one cause of autism. It's very multifaceted. It's a combination of environmental factors as well as genetics. And as a reminder, what are genetics? Well, the genetic code that is in all of our cells translates DNA into RNA to protein, and proteins are the chemicals in our body that carry out all of our bodily functions. So if something's wrong with the genetic code, it can lead to something being wrong with the protein, which may impact the function of the body. Because I work in genetics, I'm going to be focusing on the genetic factors. And there have been so many studies over the past decade that have tried to look at the genetic pattern of autism, and there's a good reason for that, because we've known that from twin studies, autism has a very high heritability component, almost 70 to 90%. At the same time, there's not 1 or 2 or even 100 genes that may cause autism. It's much more. There's a concept in genetics called penetrance, and it denotes how likely that genetic variant is to cause a certain disease or contribute to a certain disease. And autism is associated with a lot of genes, some of which have very high penetrance, like FMR1, for example, for Fragile X syndrome, MECP2 for Rett syndrome, or TC1-2 for tuberous sclerosis, which are very well-known syndromic forms of autism. And then we have the much more common strongly penetrant single gene variants, rare variants, and common variants that may not directly cause autism, but contribute to the overall symptomatology. So that maybe with the right combination of environmental factors, autism can be triggered in the person who has genetic variants. How do we know if a patient has the genetic variants that we're talking about? So there are different ways to test for the genetics of a person, and they can be from simpler to more complex. So here is how the genome is roughly organized. If we look at karyotyping, this only captures the simplest portion of the genome. If we look at micro about, it gives us a bit more information with higher resolution. And if we look at single gene testing, it can give us very precise information, but only about one gene. So that would be only this. Whole exome sequencing, on the other hand, looks at only the coding regions, so these regions of a gene, which end up being translated into the protein. But whole genome sequencing, which is the star of the show today, looks at all of the genes, so including the introns and the promoter regions and everything, for all of the genes in the body. So it's basically like a full body scan of your genetics. So how does it work? Well, in the past, sequencing the human genome was a very time-consuming, expensive, and laborious process. Today, not so much, thanks to the advent of next-generation sequencing. So usually, patients come to us with a blood sample, 2.5 milliliters, you don't need anything more. And we can take some from their parents as well to see if the mutation is present in their mother or their father. And then we do the next generation sequencing. We break the genome into small fragments, and then we create a library, and we see if what we can come up with in the analysis portion matches what we know about the typical human genome. And if it doesn't, these are the types of results that we can get. So these are all types of genetic variants that could be detected with whole genome sequencing. So this is single nucleotide variants. These are the most specific. That means just one letter of the genetic code is changed. There's insertions or deletions, which can mean a few letters of the genetic code are missing or there's some extra. It can mean that there's extra chromosomes where there shouldn't be. It can mean that there's extra letters or less letters where there shouldn't be. Or that the letters are all mixed up, as in inversions and translocations. Or that the letters continually repeat themselves. I could go on. So there's different types of mutations, but what do they actually tell us about autism and how can this be used to benefit the patient? Well, this is where the personalized treatment approach comes in. I'll present 3 case studies of patients that have come through our genetic testing service and the results that we've obtained that can then help clinicians make better, more informed decisions about their treatment plans. So this patient presented with autism spectrum disorder, epilepsy, ADHD, severe intellectual disability, and regression, behavioral developmental regression after multiple bouts of infections. They had impaired motor function, aggression, self-injury, they had neuromuscular symptoms, they had gastrointestinal symptoms and liver symptoms as well, with frequent infections, diarrhea, constipations, and they had some immune and autoimmune markers as well. They also have mitochondrial dysfunction, and if you don't know what mitochondria is the little parts of the cell that generate all of the energy that we need to thrive. So if they're not functioning, then the whole body fails to carry out its duty. So what we found is that this patient has 2 mutations in one gene called adenosine deaminase and another mutation in a gene called protocadherin 19. So these 2 mutations are interesting because they're both inherited from the parents. So this one, as we can see, is inherited from the father, whereas this one is inherited from the mother. But the father and the mother are both healthy. It could be that the presence of both of these mutations leads to the phenotype that to the clinical manifestations that we've observed in this patient. The second gene is also inherited from the mother, and this could be potentially one of the examples where a gene has low or variable penetrance, so that in the mother, it didn't contribute to an autism phenotype, But in the child, due to a combination of other genetic factors, maybe this additional mutation or maybe some environmental factors, it could produce clinical manifestations. So what do these two genes actually do, and how can we fix their dysfunction? Now, this adenosine deaminase gene encodes an enzyme, which is a molecule that helps drive chemical reactions. And this one is specifically involved in metabolism and immune functions. And its deficiency can lead to severe combination immunodeficiency syndrome, as well as recurrent infections, which lead to then the neurodevelopmental issues, because everything is linked. So what can we actually do to correct this potential deficiency or dysfunction? Well, there's already existing therapies out there that we wouldn't have known about, and this patient's clinical team wouldn't have known about had they not done the genetic testing. So there's enzyme replacement therapy. There's also a specific type of stem cell transplantation that could work. There's even a gene therapy called Stringvelis. It's lentivirus-based, which is very rare for a gene to have its own gene therapy. And there are also some drugs that are involved in this metabolism pathway that can be used to better the situation. So these are all unconventional options that wouldn't have otherwise been considered for this patient, and probably wouldn't work in any other patient that didn't have these mutations and the clinical manifestations that go along with them. The other mutation is something that's involved in keeping the skin cells of our gastrointestinal lining together, close in communication. So it's also involved in other types of cells in keeping close communication. And it's involved in synaptic connectivity and GABA receptor regulation. So if you remember, I mentioned GABA before when I was talking about bumetanide. So what kind of therapies can be prescribed for this? Well, a simple one, very simple one, is a ketogenic diet. Another one is a GABA modulator, which is in trials. And another one are stereotherapies or clobazam potassium bromide. That being said, for patient 2, we have, you will see similar but different symptoms. So we also have regression. We have autism, hyperactivity, elopement, that just means they like to run away. We have stimming, so though that characteristic autism-like repetitive behavior. We have seizures, although we have not confirmed them, and there's also encephalopathy. There's obsessive focus, so some OCD-like symptoms there, and they also avoid eye contact, so this is much more typical of ASD. But from the gastrointestinal side, they have constipation, abdominal pain, and they're sensitive to dairy products. They also have insomnia-like symptoms, and it is thought that they have, again, mitochondrial dysfunction, but this was improved with specific therapies. So this one has 2 mutations, and they're both inherited from the mother. And again, this is when we're talking about variable penetrance, where the mother has these mutations as well, but doesn't have the phenotype, and the child has these mutations, which may contribute to the clinical phenotype that we see. So why do we think that? Well, the first gene is called cycle-dependent kinase like 5, and it's involved in cell cycle modulation in general, but more specifically related to autism. It's involved in synapse formation, dendritic growth, vesicle recycling, and as well mitochondrial dysfunction. And there's a specific disorder that goes with its improper functioning in the body, and that's CDKL5 deficiency disorder, which can lead to seizures, intellectual disability, and motor deficits. So there is actually an FDA-approved therapy for this disorder. There's also some alternative methods that could potentially be considered as adjunctive therapies, like cannabidiol and a ketogenic diet, and there's some experimental therapies as well, like the insulin growth factor 1 supplementation or an AAV vector gene therapy as well. In the other mutation, which is a potassium channel, a specific type of potassium channel, regulates signaling in the brain. So again, we talk about GABA because the imbalance between excitatory and inhibitory signaling in the brains of ASD patients is thought to be a very common part of the pathophysiology of the disorder. So the therapies for this, if it's a gain of function variant, which means that the channel is more active than it should be, one could treat it with fluoxetine or norfluoxetine, which is a channel blocker. And then if it's a loss of function variant, which means that the channel is less active than it should be, you can treat it with the channel activators. And these are some experimental drugs that have been suggested. The question is, how would you know if it's a gain of function or a loss of function mutation? But we'll get to that later. So the third patient, and last patient in this presentation overall, has autism encephalopathy, poor fine motor skills, suspected cerebral hypoplasia that needs to be confirmed, fatigue, decrease in muscle look, decrease in DTRs, suspected subclinical seizures, which means that they don't have the typical seizure manifestation, but they do have some abnormal forms in the EEG, electroencephalogram. It's a brain scan that can see waves, and they have variable moods and non-verbal episodes. From the GI and the ear, nose, and throat symptoms, they have dysbiosis with increased yeast and decreased butyrate producers and protozoa. They have bloating, they have an intolerance to gluten, and it is thought that they might have intolerance to dairy or lactose as well. They also have done some previous genetic testing which showed that they have some metabolic polymorphisms. That just means that the genes that metabolize, that are involved in the metabolism of this right here is they're different from the regular, from the typical consensus sequence. And yeah, they also have elevated B12, and from what we see here, we have again a similar case where the mutation was inherited from the father and from the mother, but there are two different mutations in the same gene. So this gene is actually, again, an enzyme which is critical for GPI-anchored proteins, and these impact development, synapse formation, signaling, and the deficiencies associated with intellectual disability, hypothonia, seizures, and cerebellar atrophy. So, as you can see, it kind of matches the symptoms of the clinical manifestations of this patient. And the potential therapies for this would be to supplement with vitamin B6 and symptomatically manage the seizures as well and support the gut microbiome. So these are just a few examples, and I'm kind of regretting choosing them now because they're all examples of inherited mutations, whereas de novo variants, or the ones that are spontaneously generated in the patients, are the ones that are really interesting and thought to play a greater role in autism. But stay in touch so that you'll see the upcoming publications of those later. But yeah, we've done this kind of testing for many patients across the whole world. We've had patients from over 30 different countries, not Turkey though. So, and these are some other genes where we found mutations mutations, and from our analysis, not necessarily of the genes that are listed here, we found that these are some common, that these genes that we keep finding mutations in are involved in some common converging pathways, biomolecular, biochemical pathways in the body, so they might point hypothetically to a cohesive autism mechanism. This is just an example of one of the genes that we detected in a few of our patients, called SHANK3, which is one of the more popular genes associated with autism spectrum disorder. And while it's thought to be low penetrance, there is a specific subset of patients which are diagnosed with Phelan-McDermid syndrome, for which it is very causal. So this company called Jaguar is actually piloting a gene therapy starting in January 2024. So it actually, the results will be published after June 2031. So in about 6 years, we'll find out if it works. But it's a good example of what might come out of understanding the genetics of autism patients better on a more system level. So with the case studies, we saw what it can do for individual patients. But if more and more individual patients are tested, then the whole autism cohort can get better characterized. And I've put this here as a bit of an inspiration of what I would ideally consider doing before prescribing any of the medications, but it's a bit far-fetched. So after detecting certain mutations, we can model that in vitro systems. So that means either in cells in culture or in mouse models or in little brains that we grow in petri dishes in the lab, so that we find out how exactly the mutation detected in the patient impacts the overall function, so that then we can personalize the therapy approaches for everyone. And it would be most beneficial, I think, to pivot this move from a one-treatment-fits-all model to a more personalized diagnostics and more personalized therapy approaches, so that every child with autism gets the proper care that they need for their core symptoms, as well as their oftentimes debilitating core comorbid issues. If we can find and better characterize the different types of autism that exist from a biological perspective, then we can start to make better autism subtypes, better designs for clinical trials, and more streamlined and focused therapies with fewer side effects and better outcomes for the patients who at the end of the day matter the most. So autism is not a one-size-fits disorder, and it's time, I think, to move beyond a one-size-fits solution. Thank you very much for your attention, and I'm open to any questions you may have.
Ben sizden gün söyleyeceğim. Ama siz söyleyin. Sorusu tekrar sorun. Beyefendi çevirecek.
Bu deneyler bizim otizmli çocuklarımızın üzerinde mi denilen testler, yoksa normal çocuklarımız veya insanlar üzerindeki testler üzerinden mi tespit ediliyor?
experiments?
En son laboratuvar çalışmalarındaki
Right, so I think you might be referring to the Jaguar gene therapy. Right, so that is, it's enrolling in a clinical trial, and when it's, right, and I assume it's first, with gene therapies, it's common to first put them in the diseased people to see if they work. So the studies are not done yet, but they will be done on autistic children. And for the top study that was on the slide just about that, that was in a mouse model.
Thank you.
Ben bir şey sormak istiyorum. Dinleyemedim gerçi ama ilişkinde konularını incelemiştim. Teşekkür ederim öncelikle. Sorumuz için dinleyemedim çok fazla ama sağ olun. İncelemiştim burada. İkinci çocukta olasılık, otizmli olma olasılığını ben merak ediyorum. Onu açıklayabilirse memnun olurum.
Right, so as a lot of questions in medicine, I'm going to have to answer it depends. It depends if the child has autism due to a genetic component or if it's something else. If you really want to know, there are tests that can be done before conceiving a second child to determine the risk of having a child with a similar disorder, if the cause is genetic.
Anladım da onu biliyorum da sadece olasılık %kaç yükseliyor? Birinci çocuk otizmli, ikinci çocuğun otizmli olma olasılığını soruyor. Birinci çocuk eğer bir kişi mesela ben doğurdum bir otizmli çocuk, ikinci çocuğun otizmli olma olasılığı ne kadar artıyor diye sordum. Tamam şeylerden de bunu bilinebiliyordur diye düşünüyorum en azından oran olarak. Artış ne kadar yani ikinci çocukta olma olasılığı?
I cannot give you a percentage because your family is not like any other family. I can give you some statistics about twins, but it can be the case that your child, your second child, is completely normotypical. That often happens. It can also be the case that they might have autism or a slightly different condition, maybe milder, maybe more severe. Is the child born? Or are you planning a second child? Okay. Well, if your friend's child is born, then maybe just wait for the warning signals and be careful of early diagnostics. And if it's not born, if it's really a concern, then they can do a genetic test and get an actual answer, specific for them. So the next presenter will be Dr. Richard Fry. He's a worldwide esteemed leader in the field of autism, specifically in the field of mitochondrial dysfunction, folate metabolism, cerebral folate deficiency. He discovered actually the whole folate story behind autism, so it will be very interesting to hear. He's been involved in a lot of different autism studies. He has over 300 published peer-reviewed books and chapters. He's currently based at the University of Phoenix, Arizona. And he's done his education as well at a variety of top institutions,